Good morning, everyone. On behalf of the Department of Physics, Tripura University, I welcome you all to today's distinguished lecture program. Today's lecture will be delivered by Professor Biman Bagchi, IISc Bangalore. And today's program will be chaired by Professor S. Chattopadhyay, HOD, Department of Physics, Tripura University, and the technical session will be conducted by Professor Debojyoti Bhattacharjee, Senior Most Professor of this department. Department of Physics, Tripura University regularly organize seminar, conference, workshop, etc. time to time. We are grateful to Professor Bagchi. Despite of his busy schedule, scientist of his level has agreed and accepted our invitation to deliver today's talk. And I am sure that the young generation and students of this region will be highly benefited and motivated by his lecture. Again, I welcome today's speaker, Professor Biman Bagchi, and all the participants, my colleagues, students, and research scholars. So now I request Professor Debojyoti Bhattacharjee to conduct the lecture program. Sir, please.
Okay, thank you, Arshad. I am now, on behalf of the Department of Physics, I welcome Professor Bagchi and also all the distinguished audience who are present in this Google Meet program. And before start of this lecture, I am giving here a brief introduction about Professor Bagchi. And Professor Biman Bagchi, at present he is the National Science Chair of the DST SARF and Honorary Professor of the Solid State and Structural Chemistry Unit of the Indian Institute of Science, Bangalore. And he got that Bhatnagar Award in 1991, but recently he also got several other prestigious awards like this Joel Henry Hildebrand Award from American Chemical Society, elected foreign member of the American Academy of Arts and Science. Also, he got this that prestigious Humboldt Science Research Award of Alexander von Humboldt Foundation in 2019. He got it. And also he's a National Science Chair Award conferred by the Department of Science and Technology. And that most important thing is that Journal of Physical Chemistry, B, in 2015, just published one special issue of Professor Bagchi regarding his life and all his research activities. And I request everybody to go through this journal. And other thing is that he also got that Professor A.K. Bose Memorial Award. These are elected fellow of the Indian Academy of Science, elected fellow of the Indian National Academy of Science, elected fellow of the National Academy of Science, and Jagadish Shankar Memorial Lecture Award from INSA. And besides this, he was the Dean of the Science Faculty of the IISc, and from 2017 to 2019, and Empowered Committee. He was also a member of the Empowered Committee of the Department of Science and Technology, and National Task Force. He was also a member of the National Task Force of Supermodel of COVID-19. He is also at present, he is also this member. And this Academic Advisory Committee of the University Grant Commission, Chairman, SSCU, IISc, editorial boards of various highly reputed international journals. Yes.
Yeah, now we can stop, huh?
Okay, no, actually our audiences, most of them are students and they should know all these things, because otherwise they will get some.
All right, please go ahead. Yeah.
Okay, sir. And also he is the editorial board member of a large number of highly reputed journals like this Chemical Physics. He is the editorial board member of the Chemical Physics, Journal of Chemical Physics, Biophysics, and so chemical physics later, you also know how high standard of these journals are, and that theoretical chemistry, journal of physical chemistry communication, and several others. And the most important thing is that his H-index is the indicates the high quality of research, and his H-index is 74. You just compare. I am a senior professor here, my H-index is only 20. And other maximum H-index of this university is hardly 30. But here's H-index 74. You just imagine his level of research work. And total citation is 23,424 in Google Scholar citation. And he already published 477 original peer-reviewed research papers, and including these 25 major reviews. And therefore, I think that he will deliver a very good lecture, and I think that everybody will enjoy. And if you have any question, you may just give it in this chat mood. I will then he will then discuss after completion of this lecture. Thank you all, and I am now requesting Professor Bagchi to deliver his lecture.
Thank you very much, Professor Bhattacharya, and thank you, Arshad, for inviting me. And this is really, truly my pleasure to talk to you students. I always want and like to talk to students. I've never been to that the beautiful part of Europe, though many times. I once even bought a plane ticket, but I had to cancel. Once on health ground, and a few times others. But I'm very curious. I myself I grew up in near Calcutta, and so I'll certainly make it after this crisis, pandemic gone. We are doing a lot of work on pandemic, actually, on the COVID-19. We have published 2 papers, and the third one we're going to submit soon. It's a pity that I'm not going to talk on COVID-19, because that's still a little bit not very clear what is going on. And but there's a beautiful theory that you can do. This might appear to be a little contradiction in terms, to talk a beautiful theory on such a horrible thing. But that's what science is. We try to understand, and we try to look into detail, into the depth. And let me assure you, all the students, that there's a tremendous pleasure to be got. See, if you measure your life, what you want to do in life, really, at the end of the day, and you read a lot of Rabindranath, Tolstoy, and all these big people, one thing everybody has said, that you want to be pleasantly engaged. That means you have to be involved. You have to be something that will keep you involved at least your days. And other than that, you would be really you could be really too bothered, to disturbed by many other things. Now, so this pleasant engagement is what science and research gives you. Writing also gives you. So basically, that is one of the basic major thing at this rather senior age. I can give that. So when I look back, it's a very nice time. Yeah, there are lots of, as Rabindranath said, "Roeche kothin dukha roeche biche." There will be a lot of sorrow, and there will be a lot of departures, tragedies. But life will go on, and science gives you a wonderful way to live the life. Okay, and I really wish and hope that you get that chance. It's not easy to get that chance, particularly in your country. Okay, now to the talk. I'll come back to this, and I have written several books on doing science, particularly in India, and I just want to discuss them. But before that, I'll take you through a voyage. And this voyage is essentially will be general, historical. Some of my work, some of the other works of much greater people. And so let us start. So if you think a little bit, that beginning of the prehistoric days, man has been obsessed with time. This is one of the obsession that man has. Also, man has obsessed with space. we all looked into long space and long time in the beginning, because that's what we started. We could see the river flow. We can see the velocity of the river tide, and we can see the stars moving. So everything is very grand time and cosmos and grand scales. And so, but in the beginning, when we started in India, we had the interesting method called Shonatari, which is really wonderful. This unit was wink of an eye, and that turned out to be not too bad, and a measure of time. I can imagine saying that, oh well, I waited for 100 wink of the eye, but this is okay. Shonatari that was, and many processes that we study and are in physics, materials, in chemistry, like electronic coherence. That's so important in electrical conductivity, many things. Then enzyme kinetics, which is all your body, all of you have breakfast and which is getting digested by if you have milk, then there is galactosides and breaking the lactose into fructose. Every function of your body and enzyme is involved. Then we have photosynthesis, the beginning of life on planet Earth. That takes place in extra time scale. That takes place now in a femtosecond. 1 femtosecond is 10 to the power minus 15 second, where I have shown the electronic decoherence. That's a time scale also in a liquid. 2 molecules collide. There are molecular rotation, chemical dynamics like isomerization, then vibration, relaxation. So there is protein folding, the huge number of things continuously going on around us. This is through time. And there are also length scale involved, like in photosynthesis, and I'll come to that. Okay, so it was Manfred Eigen who shared the Nobel Prize in 19 chemistry, devising a method to study chemical reactions. That was the beginning, essentially, of small time. Long time was already there. We already had ways to, like sun clock or other measures, sand clock, or all the Middle Egyptians and many people had. India also had these things, Jantar Mantar. But when people turned into short time and short length scales, then things are very different. If you know, some of you might know that in before last interview, before his death, they asked Einstein that, okay, there's so much progress has been made in the world of cosmos, in understanding star, theory of gravitation, and black holes already done. What do you think? What will be the next line of progress? And that was towards the end of his last interview. Einstein remarked that next development will come inside, not outside of our body, and into small length and time scales. Very interesting. That was his kind of last comment on his last interview. Now, Feynman made a very similar comment also, and actually he pioneered much of the work later. Now, so did Einstein, as I discussed. Now, our Manfred Eigen was intrigued as a young man, he's a young 21-year-old scientist, that he read by, I think, when somebody, that there are some reactions which are too fast to measure. So then he did some beautiful stuff that we'll talk a little that in this slide. But before that, as I was saying, the ancient clocks are sun clock, then sand on the left, all of you have seen. But really great leap was taken in 1872. There is a wager, there's a bet in Palo Alto, where Stanford University is. And there is a person, Stanford, who was a mayor of Palo Alto, and he had a wager with another rich man. Those days, a lot of money. And the wager was that when a horse running does in any instant of time, all the 4 legs of the horse in the air. This is amazing, that wager that can have that on this. Now, one guy said yes, Stanford said yes, and other guy said no. So then they have to find out. So there's this photographer, Edward Muybridge, who is a genius. They got his help that if he can take photography. So what he did, he now set up 16 cameras in a different, slightly from each other, away from each other along the path, and then he added strings of different lengths to his camera. And now all these different 16 strings were attached to strings were different length. Listen very carefully. So now when the horse came to the spot, he pulled his string one after another. 16 different shutters went up at a less than a second interval. He could take 16 photographs and then make a movie out of that. That was the first movie that came from essentially what I call spectroscopy. And yes, indeed, you can see this, what was movie you were saying, the horse galloping. There are 16 quote unquote ultrafast, those days, snaps. And one of them, all the 4 legs are indeed in the air. So Stanford won the way here and the money. Now, our great, these 3 great people, Manfred Eigen, Norris, and Porter. Porter is also a man of many other things. So Eigen died recently in 93, 94. Even at 90, he was climbing the hill. Now, they have all done many different things. Now, so the way the Manfred Eigen, Norris and Porter did, they created a short pulse of energy by laser flash photolysis, and they created some radical species which then react, and they could measure the rate of the reaction. And Manfred Eigen did the temperature jump method. You might have read it in a physical chemistry, chemical kinetics. Last chapter used to be not fast chemical reactions, and they used to discuss, like in the book of Moore or Castellan, they used to discuss the work of these 2 gentlemen. What is important here, that is very important that they perturbed the system. They have to disturb the equilibrium and then study the return of the equilibrium, non-equilibrium, thus created at time equal to 0 to the equilibrium state. I repeat, you create a perturbation, and the perturbation, it goes away from equilibrium state. It returns, a system tries to come to equilibrium, and you get the you could measure the rate. So you have to perturb the rate. Just always, in order to get the mass, you remember, you have to lift the weight. So this is called the response, that we always measure the response of a system. And in order to get the response of a system, we have to perturb the system. This is a very fundamental concept in physics, chemistry, biology, anywhere. And there enters the concept of response functions, which is at the basis of physics, chemistry, any whatever high energy physics to everything. These response functions are the ones experimentalists measure and theoreticians calculate. Okay. We are continuing on spectroscopy. That means we are continuing on the time, and we are seeing that how time we are measuring becoming shorter and shorter, as Einstein indeed said. So then came in a great fortune with the laser spectroscopy, was discovered. Again, that was also, stimulated emission, spontaneous emission, the Einstein coefficients. It's amazing how somebody can do so much. Okay, now the laser spectroscopy based on that principle of stimulated emission. Now, high resolution electron microscope was done by sending electron beam and seeing the polarization and phases. You can do the electron. These are the electron spectroscopy of Syed. They got the Nobel Prize in 1999. Then laser spectroscopy developed by that time. Many people, many of them didn't get Nobel Prize, but they are very well-known people. And then it was used in what the earlier that I said, the Eigen, Porter, and Norris, they were in microsecond. 1 microsecond is 10 to the power minus 6 seconds. And now we are coming to a much faster time, much shorter time, that is in nanosecond. And how do you understand a nanosecond? One of my friends once told me, explain to Queen Elizabeth what is a nanosecond, and that we use all of that. That if the time a light takes to cross an apple, that is a nanosecond. So that tells you that how fantastic time scales we are talking. And it's really fantastic that one can measure processes at this nanosecond time scale. And it is also fantastic that much of the reactions, much of biology, many things can happen at the basic level is in this nanosecond time scale. It's amazing, which is something exactly what Einstein said, that it is, it would be like that. We did not. Nobody actually even imagined that these things will come. Ian Lee, I make Hartsback. I also made. They are wonderful gentlemen, very creative, fantastic people. A short, quick 1-minute story about Dr. Hartsback. I did PhD at Brown and went to University of Chicago, which was the #1 place in those days in theoretical chemistry and spectroscopy, physical chemistry. And I found everybody has so much better than me, have done so much more work than me. I was kind of very nervous and paranoid. So I started working and found some problem and did write it up and use a single author paper, and I sent it. And to Journal of Physical Chemistry. Single author paper, actually it turned out to be my first paper to come out. Then many other papers came from my PhD work, but they were still in the process of submission. They somehow came faster. Probably it was later they published it because very short paper. So the paper to refer his comment, one guy said, yes, publish it, and he signed his name. Other guy didn't sign, but had some criticism and negative. And there are, of course, many mistakes in English because I didn't know how to type. And so I got somebody, one of my colleague friend, to type it. And Dartley Hartsback then wrote in hand later to me, say, Biman, see, I never met him, but that time he doesn't know me. Biman, since this is your first paper, I'm going to accept it. You can see a generosity of a person. Here is an idiot, stupid Indian who has not a single publication, never met him. Here he writes a kind mail. So that he publishes, he accepts my paper because this is your first paper. Okay, so these are some of the people we are, the kind of respect we have for them. Not always, I'm not saying all of them, some of them. And then you better remember the good things. Don't remember the bad things. In life, you always remember the good things. And because that will give you inspiration on your way towards forward. Okay. Now, I two great gentlemen whom I know very well and work very well. Rudy Marcus, Nobel Prize in Chemistry. I talk quite a bit about his work, theory of electron transfer, because that's very fundamental in physics, chemistry, biology, material science, anything you name it is electron transfer. Another is my colleague and a genius, is as well a transition state. He took the electron transfer actually in the picosecond domain. That's why I put it here. The person who measured rate now, not only picosecond, jumped to femtosecond, was Swell. An amazing experiment he did. He did it in Caltech and not too far from Stanford, California. And he used a technique of time delay and similar things, which is the Edward Muybridge experiment. Actually, whole of spectroscopy in laser spectroscopy essentially similar things. You do a time delay by using different lenses. You split the frequency by using prism. So there is a basic thread through all our analysis. So he wrote a beautiful book, Voyage Through Time, and some of them are okay. Some of this are a little bit, too bombastic, but still is worth reading the book. Part of the title of this talk is taken from Voyage Through Time, Ahmed Zuhair. So this is from Ahmed Zuhair's book. So Ahmed Zuhair was given Nobel Prize to create the transition state. And what is so unique about Rudy Marcus' work, he identified what is the transition state in an electron transfer reaction. It is a very different reaction. It is not like a bond breaking. Whenever you talk of chemical reaction or any process, we think of a bond breaking. No. Here the transition state is created by the solvent. So it's a collective thing. It is a fully physics that one needed to describe a very elementary chemical and biological process. Now, this concept of transition state is something what Ahmed Zuhair described very nicely in his book. He said, okay, let me take a cat, poor cat, it's quite blissfully sitting there and doesn't know what is going to come to him. So he took by his tail and just dropped it. Now the cat falling, and now whenever you throw a cat, it is really amazing. It lands on his foreleg. So it rotates in space. So now you can see the fourth from the top can be considered as the transition state. That's in the middle. So initial state is this happy cat on the branch, and below it's on the floor. So it's called evolution of the dynamical state of the system. And the intermediate state is which is neither state A nor state B, is called the transition state. So this is the kind of definition of transition state that comes in many cases. We have done now the voyage through time. We'll come back again. Now I want to take you through voyage through length. There we start in the electrons, which are sub-angstrom. Then we go to molecule, like we have 3 molecules there. Water, H2O, the yellow one is oxygen, blue one is hydrogen's bond. So they are of the scale. Water, for example, the water diameter is 2.5 2 to 2.5 angstrom. Then you have a unit cell of a lattice that goes to about 10 angstrom, about 10 nanometer, a little bit more than nanometer. Then you get grains and crystals, which are hundreds of nanometers. Then you go to micrometer with a microstructure of solid, and like that. Then I go to battery and these things, which are macroscopic, or your pen or pencil, they are macroscopic. However, they are all built from this. The interesting thing is that at the time of Einstein's interview, the electronic part was quite well understood, and people are already starting to do really sophisticated theories of conductivity. Though it happened after Einstein, but 1958 was the famous Phil Anderson's paper of electron localization. And 1961, 62, and that time was the famous Leon Cooper pair led to theory of superconductivity. And so microscopic aspects of space was somewhat better understood and resolved than time. So now let's get through time. It also took time to come to this stage. One most interesting and hilarious thing is our great philosopher Aristotle. I'm a great admirer of Aristotle. There are books you go to any good bookstores in a university, you will find racks and racks of things on Aristotle. These days, only philosophers seem to read books. Okay. Now, the Aristotle, this is some 400 BC or so. And Aristotle also the one who tutored Alexander the Great. Aristotle is the one who gave the map of the world to Alexander and told him what to conquer everybody. Whatever it is, Aristotle had school and a great number of students, but he had always this, he was considered one of the pioneers in experimental physics or experimental science. And he had this image, this a picture of the universe. Earth in the middle. I think you are still influenced by Bible. No, he said the Bible, sorry, because Bible came later. So, but you look at what is important, I want to tell you, is that he has a separation, he has a length scale. He correctly put he got the sun wrong, but otherwise he has put the moon next to Earth, so he has a length scale, he has got some of the things correct. Okay, but important thing is the length scale, macroscopic length scale. He also made the maps. He's credited with making some of the earlier maps of the world, Aristotle. He had one more thing, wonderful thing, he got it wrong, that he was he somewhat correctly connected that brain size with the number of teeth. But he came to the conclusion that man has 32 teeth and women has 28. So there are hilarious things that Aristotle did, but he thought about the problems. So he raised the questions. For example, size of the brain, can I connect it to the teeth? Okay. Then Zacharias Janssen, the fast compound microscope using 2 lenses. And that is really pioneering work, right? In 1590. Look at that. That was the René Chataille. you know, full-blown renaissance. Then 1675, the first man to make an user-real microscope in study of things. That was done a Dutch scientist. This was much of the activity was going in Holland and those places. But we are going now slowly, shorter and shorter lens scale. First ultra microscope was using many lenses, still optical microscope, and studying objects below the wavelength of light was that wonderful thing by these Zitsmongi and of Zarnicke, who did many other things, phase contrast microscope, that he realized that it is not just the brightness, I can use the phase to, and here the first time one could see inside a cell. And there are beautiful pictures in Google. You can see that here is a brightness picture by just intensity. You can see nothing. But here you can see the phase contrast microscope, where you pick out the phase interference and destructive interference, and you can figure out the thing. There's a beautiful picture in internet on this phase contrast microscope. First electron microscope using electrons beam was done by Ruska, Nobel Prize 1986. So we are now going to the length scale of a nanometer and below. And then scanning tunneling microscope, which made these two right side, TEM electron microscope went into scanning probe microscope, and tunneling TEM, they kind of ushered in the revolution in material science. And very short, very short length scale. Now, after what happened is this, I'll have a better slide on the single molecule spectroscopy, but just this is something I think, bear with me for a minute. So now, there is, where is this? So we have developed time through spectroscopy. We have developed understanding of length through microscopy. Now you'd ask that I want to combine the two. I want to know how microscopic length scales are changing on a microscopic time scale. And that has been done through single molecule spectroscopy, which already got a Nobel Prize, might get more Nobel Prize. The single molecule spectroscopy combines the spectroscopy and microscopy. That is essentially the meaning of single molecule spectroscopy. Very powerful technique. Now, even in India, when I first used to give talk on single molecule spectroscopy, that was more than 20 years ago. And that is as the only person in India was talking of single molecule spectroscopy, and I was giving theoretical talks. Of course, I'm a theoretician, though I am very close with experimentalists, and many of my colleagues are experimentalists actually. And but now, and actually from 2000 away, there are a lot of, because it's very expensive. But now many people are doing single molecule spectroscopy and getting wonderful results. Like Professor Pankaj Bhattacharya did a lot in cultivation of science. Then other people are doing in many different parts of India now, but it's still very expensive. And so a combination of this, as I said, spectroscopy and microscopy is the single molecule spectroscopy. I'll have a lot more to talk on that. Okay, then also because of this combination, we can like ultrafast X-ray spectroscopy, something I did not talk on. I should have talked on ultrafast X-ray spectroscopy. That is being used to do bond-breaking process. You know, it's amazing. Then you can do how molecules move. Isomer is a very important biological process. Like the reason you can see is that when you open your eye, there is a proton pump that starts, and that is initiated by isomerization in a halobacterial membrane in a polymer at the C13 position. There is an isomerization that switches on the pump. That's why you can see. It's a very fundamental that takes place in 200 femtoseconds. That has been figured out. So many things have been figured out, and it has been amazing amount of studies that I'd say that computer simulation and computers, laser spectroscopy, microscopy, all these things kind of came together in 1980s. So starting from mid-1980s to all the way 1990s and 2010 or even now, amazing kind of festival going on in this area. Huge amount of things, many different areas, but there is a way to view them. The way as a student, you need to view them as a more or less what is the unifying principle, what is the important thing that is going on? Well, you know, they might be diverse, but there is always unity in the diversity. You know, that is the usual philosophy that Rabindranath and Mahatma Gandhi all promulgated, that the diversity, India is the unity in this diversity. And so in science also, there is a very few basic principles. You have to understand that. Manifestations and applications can be wide. Okay. So I have now talked theory. I have talked about experiment, spectroscopy. I have talked mostly of the microscopy. I have not talked of theory at all. So I'll spend 3-4 slides now to tell you a little bit about theory. You all know quantum mechanics, and we do quantum mechanics. And so there are two pillars, statistical mechanics and quantum mechanics. Quantum mechanics, most of you know, Schrodinger equation and similar things. Statistical mechanics is something that it is not taught. Even in physics, when I give talks, many times physics young assistant professors come and tell me, Syed, when you tell statistical mechanics, we understand it, but we never understood when you were taught in our classes. So I have removed some of the slides to go so that I can go far, but I will discuss a little bit. 3 minutes on statistical mechanics. I want you guys to be aware of this subject. As I said, theoretical physics and theoretical chemistry or theoretical biology, theoretical material science, all based on two things. One is quantum mechanics and this statistical mechanics. You can say these are the pillar of two pillars of theory. Whole theory is resting on these two. Now, statistical mechanics, as the name says, statistical, which is probability, mechanics, you know, classical mechanics, the Newtonian mechanics or Hamiltonian mechanics. So conversion of the statistical mechanics is mandated to convert microscopic information to macroscopic observables so that we can explain phenomena. So we start with intermolecular interactions en route to calculate entropy, free energy, viscosity, rates, why things happen, how things happen. So it is why and how in the macroscopic world. I repeat, why and how are the two important questions, and they are explained by statistical mechanics by using quantum mechanics. Quantum mechanics is an in study of statistical mechanics. How did statistical mechanics started? I must spend some time on that because I already spent enough time on experimental science. So let me spend a little time on the concept. And you know most of it, that the James Clerk Maxwell formulated the Maxwell distribution, e to the power minus mv square by 2 kVt, remember? That was the first ever statistical law. Before that, people were using really mechanistic law, classical mechanics, Newton's equation. So everything is completely deterministic. You give me that time t equals your position and velocity. I give you position and velocity of the particle at time, later time t, by using Newton's equation, or in a little bit more complicated because I can use Hamilton's equation. However, that doesn't help me. That doesn't help me to explain, understand my body. That doesn't help me to understand how COVID-19, the RNA and the virus is working. And it doesn't explain me why water is what is water. That is why we live on water. Why water is so important. What is so peculiarity of water? Doesn't help me. Why? Because if I want to go and do each molecule, each time, step and all these things, I get a huge amount of information which cannot make any head or tail of. Too much information and without any kind of rationalization. Statistical mechanics allows you that. Takes the information, takes in the information, go to a reduced description. And building up the reduced description is the name of statistical mechanics. That from Avogadro number of 10 to the power 23 particles, how do I get to this, say that I can describe viscosity of water as 1 centipoise? And pressure is this. How do I do law of corresponding states? Why PV equal to nRT? And all these things, equipartition theorem, how do I understand them? How do they help? This is what statistical mechanics does. Now, James Clerk Maxwell wrote these 2-3 beautiful papers. Actually, somebody in France did it before. But it was done probably bits and pieces. Maxwell put it beautifully in 2-3 papers. One of them was the last paper, became our great Ludwig Boltzmann, became obsessed with that paper. He always carried that paper with him till his death in 1906. She's the father of statistical mechanics, and he really introduced deliberately and systematically the concept of probability into mechanics. That's why he's considered the father of statistical mechanics. He derived the famous formula A is equal to KBL omega. You know, he did many other things. He worked what we call microcanonical ensemble. On the other side of Atlantic, there's a great man, Willard Gibbs, whom Einstein, when Einstein was asked who is the biggest thing you, Einstein said, I never met Maxwell, but I never met Willard Gibbs, but had I met him, probably he's the one who is the most logical mind. You know, he placed him above all other great people. You know that Einstein's time. Now, according to Robert Mulliken, he did for statistical mechanics and thermodynamics what Laplace did for celestial mechanics and Maxwell did for electrodynamics, namely made his field well-nigh in his theoretical structure. So what did Gibbs did? He introduced two postulates. Somebody please turn keep your mic mute. Okay. So two postulates he did. One is time average equal to ensemble average. First he introduced the concept of ensemble, which is a beautiful concept. And he said, okay, what I have to do, I have to create a mental replica of my system, which is characterized by a constant number of particles n, constant volume d, and constant e. I create millions and millions and trillions of this replica. Then I can say if the system is at equilibrium, then each of my system will be in a different microscopic state. Why? Because the number of microscopic states is even larger, much larger. 10 to the 33 in a particle in a box in 1 cc. That many 10 to the 33 microscopic states. So each of your mental replica will be in a different microscopic states. Now, when I do average in our area, we say time average, which is given in the first, that I measure something for a long time and then average by the time. That's a time average. Maxwell said I can equally do by ensemble average. So I'll average over my ensembles. And he assumed that the ensemble average will time average. Brilliant. I think I consider ensemble and the average is one of the most, will be probably top 3 all-time brilliant creation of human mind. So once he did, then he said, okay, now I can calculate, of course, the ensemble, and I can, but I have to do ensemble. I need to give a probability to each my member of my ensemble. Then he said, I know nothing, I know except all of them are equal energy. So he said equally probability. That is, all of them are equally probable. But then he had landed in trouble. Okay, but I have to now go back to time average and I have to make that my time trajectory is long enough that it goes through all the microscopic states. That is the ergodic hypothesis, which guarantees that it is I can building a system. I'm assuming a system which goes through all the states of the system. So these are 3 brilliant things of time average was done by Boltzmann. Ensemble was introduced by Gibbs and did this, and that is the essentially basic of statistical mechanics. This was done just around 1900. Then enters the person who puts many things in dynamics into right cases. So, as I already said, in Manfred Eigen and all these people, that rate, chemical reaction, chemical change is a fluctuation of equilibrium. It turns out what Einstein did in several masterpieces on a 2 disjointing, people don't know that they are connected. One is a Brownian motion, which was mentioned in his Nobel Prize also. But another thing he did, he developed a thermodynamic theory of fluctuation. And he did that, okay, specific heat, nothing but the energy fluctuation. Now he sits so well with Boltzmann and Gibbs that if I can study fluctuation of energy of a system, which is now in a heat bath, temperature is constant, but so energy is exchanging energy in the bath. I do the fluctuation as when this energy is going up and down, I take an average, and then delta is the dispersion, and I take square an average of that, then that is the specific heat. Can you imagine? One of the most important response function how much heat I need to increase temperature of a system by 1 degree specifically. That is nothing but how much energy spectrum is there. That is delta E square. Similarly, your isothermal compressibility is fluctuation volume. It goes over to dynamics also. Your fluctuation total dipole moment is a dielectric constant. This is the wonderful. That's why the response functions are the most important thing in physics, biology. Everybody calculates response functions in physics and Cramer's chronic relations. Some of you know, some of you might not know. So one thing before that is 1907 was thermodynamic response function, but 1903 to 1905 he developed what is the Brownian diffusion, because this was a Robert Brown who was looking at the microscope and a pollen on a beaker of water, on surface of water, and that was always moving. That was the time, you see, microscope was just available, optical microscope. As I told you, the many things kind of happen in science together. So he was looking at the microscope and found the pollen is constantly moving, incessant movement. Then he was very surprised. He said, these guys have a life of their own. Initial thought was that they're living. But then he took dry leaves. He knew not that also moving. So when this Einstein came to know of the experiment, he immediately formulated what is the beginning of time-dependent statistical mechanics. Not quite beginning was Boltzmann and Maxwell. So third important contribution, the Brownian diffusion. There he said a particle constantly moves. It executes a random walk. And I can see mean square displacement and time averaging. I can get the diffusion, which is this, both the two goes by the name of Einstein's. One is Einstein's formula and Einstein's equation. The diffusion in terms of mean square displacement, and diffusion is connected to a property of the solvent now, which is viscosity. The zeta is friction. And here is some, I hope you can see that a molecule simulated in a liquid, two molecules, and see they are completely going around. This is the mimicking the pollen in a much faster time scale on water. So this is the random walk, continuous. This is called drunkard's walk. This is then went on to actual Langevin equation was already written down by Einstein, but without the fourth term. So V is the velocity, M is the mass, zeta is the friction, F is the external force, and R is the random. So what we have here in the R0 RT in this case is the dynamical version of the linear response theory. These are beautiful stuff. You read up. There are many books. You can just these days you can do Wikipedia. Think of random walk. Think of Langevin equation, and you will be able to do. Jump a little bit more, and I'll come back and forth that this transition state. So there's a lot of study. These are Nobel Prize winning work, 1956. A lot of study how electron transfer takes place, which is a very important chemistry and biology. Photosynthesis, for example, which was done later and did get a Nobel Prize. But in ordinary redox reaction in chemistry or battery electrodes in material science and physics, you have the electron transfer. But how does electron transfer takes place? How does electron go from one molecule to another molecule? What Rudy Marcus pointed out that it does not go because there is no chemical bond. Many of the time, no chemical bond is broken or formed. But electron goes. I have given this DNA in water, and I have given the water as a dipole moment as an arrow. What happens by spontaneous fluctuation? Water molecules arrange themselves such that it comes to a state where electron doesn't know where to go, to D or A, and it goes from one to the other. So dA becomes D plus and A minus. Electron transfer takes place. But it is done by surrounding solvent. It's a beautiful phenomenon. That's why a single Nobel Prize was given. Beautiful phenomena where the reaction coordinate is a collective reaction coordinate, which is the polarization of the surrounding medium. So then this theory that was worked out by the great man is, and I give this theory simply because that how power of simple algebra can be important. So here reactant is one harmonic surface, reaction coordinate is X, which is polarization, which is the sum of the dipole orientation of the dipole moments. And then when going to reactant to product, both are harmonic surfaces, and the harmonic surfaces cross. Where they cross is the activation energy, the energy needed to go there, which could be calculated by using dialectic continuum theories, as it did. Though you don't have to go through the equations, but you have to appreciate the fact that we are describing the movement, collective movement of the solvent, polarization as a harmonic surface. And there's a force constant. And the force constant is characteristics of the solvent. And this is so the reduction that I was talking before, the statistical mechanics of Maxwell, Boltzmann, and Gibbs. The reduction in degrees of freedom, I am not considering all the water molecules. I'm adding their orientation, and that gives me a polarization. And I'm following the polarization as a particle. So this is the reduction in degrees of freedom of Avogadro number. 10 to the power 23 particles are reading in 1 or 2 degrees of freedom. This is of course a fortunate case. But this reduction in degrees of freedom is the hallmark of statistical mechanics. That's what statistical mechanics does. That it takes an enormous number of degrees of freedom and then reduces to some comprehensible that a human mind can calculate, human mind can understand. So reduction in degrees of freedom is a common theme through this talk. Response functions of Einstein's is another common theme through this talk. What he predicted, some beautiful thing he predicted, okay, if I make this delta G, which is stability of reactant and product, that I can do by doing varying some groups and varying the electron affinity and ionization potential, then I can do the delta G. Then he predicted that delta G function of that, people before thought that if delta G increases, rate becomes faster, the product becomes more stable. Rudy said no, it's the other way around. Beyond a point doesn't help. And it goes to a maximum called Marcus parabola. And it's very interesting that the product is becoming more stable, but beyond a point, rate becoming start decreasing. Okay, because of this thing that we already said, that when the delta G increases, then this product curve, harmonic curve goes down. Then it intersects where there is no barrier, but then again it goes up barrier. So from this simple diagram, you can understand this far-reaching result, which was indeed by the University of Chicago. It was done when I went to University of Chicago. The work was just getting done, which finally gave Rudy Marcus the Nobel Prize. Before then, Taube was given, and Rudy was extremely angry. And during my stay in Chicago, Henry Taube and Rudy Marcus both gave talk there, as one chemist day or physicist day, like you guys are doing. Two great people gave talk. That year, Taube got the Nobel Prize. They thought both of them will get it together, but not giving Marcus, Nobel Committee had to make up by giving another single Nobel. To my mind, one Nobel was wasted. But this Caldera, Leggett, and Claus famous experiment actually showed that how correct Marcus was. Marcus predicted 1956. It was verified in 1984. Really, it is a triumph of theory and statistical mechanics. Okay, so electron transfer reaction find used in chemistry, biology, photosynthesis, many other material science, batteries, electrodes. That's what I spent so much time. Now, he is my colleague and friend. Many papers with Graham Fleming. Graham said to find out, okay, if I want to do electron transfer reaction, I need to know how fast the solvent polarization moves. And he did this experiment where exciting the molecule to a higher excited state, which has different polarization, different charge density, different dipole moment. So stabilization is different. Now, those days, femtosecond laser has come. So Graham could measure the shift in the spectrum, and that turned out to be amazing. There are three components in water. One is 70 femtoseconds, and this is about 240 femtoseconds, and this is 1 picosecond. One, we did not imagine water being water. We thought water is a little slow, so hydrogen bonded. No, polarization in water is extraordinarily fast. Nobody thought it's possible. He got every award other than Nobel Prize. It's amazing that water is so fast. Water also turned out not only the most convenient, most liquid, but also the fastest liquid. Even remember, water should not be a liquid after all. You know, it is very small molecule. All of them much heavier molecular gas at room temperature, ambient. But water, because of hydrogen bonding. So we all thought hydrogen bonding. Water rotates slowly, about 10 picoseconds. But when it collectively adds up, that this collective order parameter, collective response that I'm talking of, that is ultrafast. That happens in a this sub-100 femtosecond time scale, 60-70%. This is amazing, amazing discovery. At the same time, when lasers came, we started doing a lot of beautiful work. We started looking into chemical reactions, which are very, very fast, ultra fast, the kind of things Manfred Eigen could only dream of. So we could now have many different molecules like malachite green. Those of you who have done titration, malachite green, ethyl violet, crystal violet, and all those molecules, and many other dyes. They all undergo these kind of very fast reactions called electronic glaxation. That electronic glaxation is picosecond, 10 to the minus 12 second, and we could now measure that. We could measure that, and some beautiful theories, like there's a famous theory by Kramers and Kramers' old 1940 theory that kind of broke down. I'm sorry about this, did not come up. I think it is, I'm using a new computer, math type still not installed. But this is a fractional viscosity dependence. So the black line was the theory before by Kramers. Experiment gives the red line. Again, Fleming-Hochstrasser, and theory can explain this thing by doing something really very deep, saying that when you have a barrier, a very fast barrier, then this is what happens. You know, you don't probe the all motions of the solvent. You probe only the fast motions of the solvent. So fast reaction couples to fast motions of the solvent. Similar electron transfer reaction. That's why electron transfer reaction also is so fast. It does not get retarded by slow motion. Yeah, it is a very nice concept. So the way the experiment was done, this was actually, I showed this kind of historical. This was in front of me and drawn by great Amit Zuel himself. This is a spectroscopy, the experimentalist. So look at the color code that when he excites, you excite by using the violet or ultraviolet, then it shifts towards red as system comes down on the excited state, and the red ultimately. So this is solvent is dynamic, and energy is coming down, and there's a Stokes shift to the red, and that is measured by Graham Fleming, Amit Zuel, and other people. Okay, now I'll switch the gear, talk a little bit of biology. Again, the same length and scale, length and time scales. It's a very important problem, protein folding, understanding through chemical kinetics and similar things. So how the protein folds. So protein, let us consider when it's created, it is as a nucleus, it is kind of those tRNA, mRNA, and all the, it is kind of unfolded, disorganized. But very quickly it goes to a folded state. What is so interesting about that, a protein in your body, my body, everywhere, same protein is the same unique native state. That's amazing. That how does it find the same state every time? So many trillions, zillions of time in everybody's day. And many of them are even preserved to evolution. That means if they are even in animal bodies. What is so uniqueness? It's just mind-boggling, because protein is not a simple thing. It's fairly complex things. So protein problem is essentially a problem of predetermined contact pair formation. This is very important. Very important. Why? Because it has to form the same contacts. And I'll be a little bit more on that. So note that different from liquid solid phase transition. This is the example I always given, where some of you might have heard and read my notes on this. That if I do melting, let's take sugar, or I take sodium chloride salt and dissolve it. Then I evaporate the water. Then I can get crystal back, right? And say I have a thought experiment. I level each atom, 1 to Avogadro number. Then each time I dissolve and I recrystallize, my neighbors change. In 110 is close to maybe 88. Next time that 10 he finds is next to 1 billion something. So every time my neighboring least, nearest neighbor least changes. However, not in proteins. In protein folding problem, going to native state, every time the same contact pair formation is amazing. Is a huge entropy cost. Huge entropy cost. And that was the thing why it's so important. Topology is so important. So this is the Nobel Prize winning work of Anfinsen experiment. He did two things with the ribonuclease and also with the lysozyme, egg lysozyme. So here, what is that? Look at this beautiful thing. So ribonuclease is quite a long amino acid chain. Might be 88, 89, I forgot. But important thing to know in this kind of a ribbon diagram, there are 4 disulfide bonds between 70 to 68, 58, 110, 95, 40, and so these numbers are the numbers. Oh no, sorry, 129. Yeah, 129. I remember. Amazing is that. So one end is here, another end is there. 2 ends you can see. Now, what is amazing is that I can break these disulfide bonds, sulfur-sulfur bond. How? By using some chemical like urea and beta-Markov tunnel. And when I break that bonds, it again becomes kind of a coil, structureless. There are some secondary structures, but we are not going to get into that. Now I remove the urea. I again back exactly same ribonuclease. What is so important is that even after they again 72 bonds, there is no reason that 110 will with 38. It could probably do much better job by getting with something close to it, like 85, 95. No, it forms again the same bond and it regains its biological activity. Now think of the experiment you are doing in your classroom. There are you are all seated, 40 students or 50 students. Now I fire a cracker suddenly and tell you, oh, fire, all of you run out. Then I said, no, this is the hoax, come back. You come back, I tell you come back quickly, I don't have enough time. You will come back quickly, but you will see your nearest neighbor is changed. The 2 students who are on 2 sides of you have changed now. That is the common thing. That's what I call entropy. But not here. Here, protein folding is so important because of these contact pair formations. Same thing again and again. And yeah, it's stupendously difficult. But in your body, my body, everybody's body, monkey's body, same native state. So that was the thing. Already got 7 Nobel Prize, but we'll get more. So they asked Anfinsen, why it happens? Anfinsen said, because native state is the most stable state. Okay, native state is the most stable state. Okay, but then in order to know something is more stable, like now know that somebody is getting more salary, rich, we need to know some poor people, right? Otherwise, there is no scale. So how do I get this scale? So I have to then explore some nearest energy, so that I know it is the most stable. So Cyril Leventhal posed this beautiful thing, becomes very famous. He's a smart man, but this is what he is famous for, Leventhal paradox. He said, Let me ask, how long does it take for a protein to fold into a native state? And this is the beauty of reasoning and so simple. He said, let me consider proteins are made of amino acids, carbon backbone, and then each lays into a base. This we call amino acids. And so 100 on amino acids, each amino acid can have say 3 states. You can find it everywhere in Google. In a very interesting articles in France, everything, and it stands in English. Then there are these many configurations. Even if the protein samples 10 to the 13 per second, which is very fast by rotation of the groups, still it will take 10 to the 27 years to try them all. Now, if my native state is a stable state, first it has to go through these states to reach the native state. But even then, to know it is stable, I'm more stable than other, that I have to stay here, it has to probe certain 1% of that or 1%. That's a huge time. But no, protein folds in seconds, some in milliseconds, and 1 or 2 in microseconds. How does it happen? That is the paradox that Levington posed. Okay. So how to get the resolution of that? There are many resolutions, but one of them, which is most popular, comes from Richard Dawkins, blind watchmaker in theory. Like Richard Dawkins was a neo-Darwinian theory, very famous. You can remember of his tweet. He is very much anti-religion, theoretical biologist, perhaps one of the most famous living theoretical biologists. I'm really fond of Richard Dawkins. So there is a book by Francis Crick, the great Francis Crick, the double helix, called Blind Watchmaker. So when I started working on this problem, I was in NIH, we were working with great man, I'll show his picture later, Bob Zwanzik. And I tried to ask something, Bob. I never did any biology before. What to read? Bob did not help me at all. Disdainfully told me, no, I've never learned it, but soon you will be talking about distal histidine, proximal histidine with a lot of disdain. So Bob being no help, I went to our chief, Bill Eaton, and the famous scientist. Bill, what do I do? How do I start? Bill said, Biman, for physicists like you, you should start What Mad Pursuit by Francis Crick. He made it jokingly, but I started Nothing to Go On by Blind Watchmaker by Francis Crick. And there in chapter 3, there is this is the chapter Blind Watchmaker, which is from the book of Richard Dawkins. And there's a beautiful example. A monkey typing one line of Hamlet, sonnet, Shakespeare, may think it is like a whistle. Richard Dawkins gives the example. 28 characters, including 6 blanks. 27 possible choices, 26 letters plus 1 blank. How can I now arrange these alphabets in so many spaces into me think it is like the Shakespeare sonnet? So he said that let us consider it. Let us imagine a random attempt by monkey. He will have to 10 to the power 4 keystrokes, and one of them will be correct. But if I make the rule, the monkey cannot change those letters once correctly in space. A few thousand strokes is enough. I did that experiment. So when I read that, I was traveling bus. I knew where to take bus because there's no parking. I immediately went and said, this is the way. So they consider 2 states per bond, connecting amino acid sequence, correct to incorrect, incorrect to correct. And we did the mean first passage time. But we did one thing. Just like what monkey, once incorrect, you do not let it. We bias the correct one. So correct one is more stable. So all the incorrect bonds I can also give the correct bond 0, a number, incorrect bond 1. Those are, Ising spin. I can say Ising spin up or Ising spin down. I can sum up just like Rudy Marcus did. I define an order parameter, collective order parameter, sum of all the things. So if incorrect is 1, then ace is the number of incorrect bonds. This is the way we do an order parameter. They're amazing. And then we can do that calculation. And there's a beautiful formula emerges. This is n count on 1 plus k naught by k to the power n. And these things comes to the law of large numbers. And law of large numbers, but biased by one correct state has more correct state is preferred energetically by you. And then you can see K0 by K1 and K0 by is our physical chemistry, equilibrium constant, forward and backward reaction rate, and this all physical chemistry second year BSc we know, physicists also should know. But then just a small bias, modest preference reduces from 10 to the power 30 years to 1 second. And so this is the what also known as Monte Carlo preference, and it is everywhere, all the textbooks. Initially they used to refer, now they don't even refer. Weatherstahl, Ellinger, everywhere it is there, and also many other textbooks. So now Levinthal paradox solves the question Levinthal said, and which actually was a key to increasing our interest and our A2 protein folding. But the way it was done then later by many people, including Peter Wallines and Unuchik, is they said better way to look into it, best way to look into it as an entropy energy. So as the protein folds, it goes to lower and lower energy. It becomes enthalpically stable, but it loses entropy because it forms this contact. As I'm talking, entropy is very important. So it goes down. So they envisaged a funnel, and the funnel and the protein folding is a flow in this funnel, which is called configuration space diffusion. Essentially Einstein theory of diffusion, but in a configuration space, it's a many order parameter space. Now, this is rugged because there are traps. Once it gets in a minimum, which has a certain number of correct states, but some incorrect, it can be stable and it can be stuck there. So, however, what is very important to know, unfolded state and folded state are often the very same free energy, which is shown in the last figure on the right. Free energy almost the same. Very, very interesting. But going from one to the other, the difference in maybe 1 kilocal per mole or less. Biology is very soft. It never has anything because it has to open up. It has to unfold and fold many times. That's what our enzyme works. It's really beautiful. And the barrier is also not very large, but so much going on. So it kept people busy from 1960s onward to 2020. Protein folding is a grand problem of physics, chemistry, and biology. And there's one more. The credit is given. I've taken it from Wikipedia, that how entropy and energy or free energy, it should be written. It's not free energy, whoever did it wrong. Entropy and energy. Good that I gave the credit. So it is showing that how a kind of a coil, random coil, unfolded protein going through defined minimum state and finally going to the native state, which is the most stable, energetically most stable state. It's beautiful, but it can get stuck there. Many times protein get misfolded. When protein get misfolded or unfolded, that can cause serious disease like Alzheimer's disease, mad cow's disease, many diseases in our body are because, and when you have cancer also, the problem is that the proteins that a cancer cell develops or does are all messed up. So a cancer cell does not do the biological purpose it is supposed to do. So it is protein folding is at the root of many diseases and many phenomena in biology. That's why it is so important. Now, after let me go slightly through single molecule spectroscopy. This is a beautiful Nobel Prize winning work that was done by Moerner. What he did, he took pentacene and a layer of pentacene on paraffin wax, very dilute solution of pentacene, and then he shined light. But by that time, the spectroscopy and microscopy has gone so far. Spectroscopy, through our ability to count every single photon, through single photon counting, amazing technique. And spectroscopy and microscopy by being able to zoom on a very small region. So this combination of spectroscopy and microscopy takes to single molecule spectroscopy. And here you're seeing emission, fluorescence from a single molecule. Amazing. Not only that, you can see a molecule moving. Those who are blurred are the molecules that we have captured them in the move. Okay. So this Moerner et al. Got the Nobel Prize later, I think 2014-15 sometime. And this is Shimon Weiss, very famous They have a special issue on the single molecule spectroscopy 1999. This is a great line by Shimon Weiss. It's good to read. The ability to analyze individual molecules still amazes even the most jealous practitioners of the emerging field. When I saw it, one person, Paul Barbara, my very good friend, I don't have the work with him, but there's some very good work. It came out in Nature, but I don't have any A for that, but too many things. The introduction of novel technique generates enthusiasm among the converted, but doubts among the skeptics. And Paul showed me when he took to his lab. Come on, here, come. When I visited his lab in end of 1998, and he showed me that. I didn't believe. I said, no, it's not. Well, no, Biman, these are single molecule. These are the. He was doing MHPPV. These are the emission from a single polymer molecule. Then I did work, and how do you do that? That's very important. How do you do? Two things important. You want the probe molecule to be under study, and you have to get the light it's emitting. First, by making a concentration of very small, and so that you have this given by your zone, where you can focus on, which might be a few micrometer cube, say 10 micrometer cube. We can do now microscopy. We can zoom on that. And it will so dilute that there's only one molecule there. And then I keep the continuous laser, continuous wave laser on. And on a different angle, I collect the photons. So it's amazing that you can do it. I can do the calculations. I used to do the calculation showing you how it is done, but it's not done here. But this is it pictorially what is done. So in an ensemble picture, I already said, ensemble picture, you have this many molecules in your volume. But now, single molecule, you have much smaller, much dilute solution, and much smaller volume, so that you have only one molecule there. And emission spectrum, ensemble, I get all of them. And what I get, the thick green line, sum of all of them. I don't know what is coming from where. What is the character of that? What is the environment of that? But by single molecule, I can get single emission from a single molecule. What is the advantage? I can now know what is the environment of the molecule, who is neighbors, how many neighbors it has. So single molecule spectroscopy is a wonderful technique which gives both spectral, both space and time, resolution, length and time. One more is the single molecule scheme, how it happens, the photon detector, everything detects, photons are emitted, collection optics, and so your laser, continuous laser goes in, and the molecule is there, which is emitting, single molecule. So highly fluorescent probe is usually taken. And there are all beautiful, beautiful things that came. Space-time information, the title of the talk comes from this single molecule spectroscopy. It is done by doing an experiment called fluorescence resonance energy transfer freight. Fluorescence resonance energy transfer freight. The rate of freight has a very strong distance dependence, 1 over R6. So in blue and red are separated, long distance. If I excite red, it doesn't go to blue. However, if the other way around, okay, it's red I am exciting. Yeah, not a great thing, but okay. I exciting red. If it is far, then only red emitting. But if they are close to each other, then energy can transfer from by Coulomb exchange. Quantum mechanics tells you what is the rate. That rate is 1 over goes as distance r, 1 over r6. So now I suddenly see emission from the acceptor, which is red. Dot is the acceptor, blue is the donor. And I suddenly get a fluorescence at different wavelength, which is characteristics of the acceptor. So I know. So I can now tell the length. And I can tell how long time it takes. I can tell the time. So both time and length are coming. So this is single molecule spectroscopy with fluorescence resonance energy transfer. So SMS freight is a technique widely used all over the world. So then one of the technique that was done by solve protein DNA interaction. There's a lot of discussion. How do protein and DNA interact? You always hear protein DNA interaction. All the proteins are coded by DNA. All the water, these water ions, they play a very important role in all these interactions. Then when synthesis has to be turned off, then it's done by protein. You smoke, you have a problem. That is a protein goes, a lac repressor goes and correct guanine to oxoguanine. It's an extremely important process in biology and heavily studied. But what was really interesting is that a protein, when it has to find a DNA binding site, it finds it not only unerringly, but also very fast. Faster, much faster than ordinary theories of biochemical reaction. How does it happen? So people said it is a sliding motion because protein slides along a DNA. But if you do that, that also takes time. So what they did, it is a combination of it slides, then it jumps. A lot of beautiful theories have been done. Okay. So this is where HOG1 repressor, when you smoke, your guanine becomes oxoguanine, which is cancerous. ROC is reactive oxygen basis, and then you don't have to, but you do enough exercise and you can, you should never smoke. But in case you smoke, then your HOG repress, the HOG1 can go and make guanine into oxoguanine into guanine. That is the purpose of these things. They do many other things, of course. So I was visiting a lab at Harvard. I was teaching there as a visiting professor there, and I did some work with the lab of Sunny Z, wonderful person. He's a famous guy on single molecule spectroscopy. So he showed me that they have a microfluidic, where the protein microfluidic arrangement, where the DNA is elongated, and he can see through single molecule spectroscopy the protein is moving, and he can get that from there the diffusion constant of the protein. So now the diffusion constant can tell me that what is the mode of diffusion. So that was very interesting, and these are the different experiments he did, pH dependence and all, many dependence, and you can see mean square displacement is linear, which means there's a diffusion, and it depends on many conditions. So now the question is that how does it go? One is that it is like these two movies made by my student long time ago, maybe Biman Jan, I don't know who made this. Okay. So there is a random motion, but there is a restricted motion. That means restricted motion means it's following a helix. It can go much faster if it follows a helix. And however, anyway, this is a interesting thing. So one can do, the theoretical interest is that we know the motion of helix. So it's a parametric equation. And I can derive this beautiful equation for to the friction. Then you have Einstein's equation, D equal to kVt by zeta. And what you find that is the agreement is just, I don't have the figure. The agreement is beautiful. So it is now established that a protein moves along helix in DNA. It is not random, but it sticks to a helix and helical patch. So it kind of zips around, and the beautiful motion. Not 100%, you will not like that, but it's. I have been referring to now and then the computer simulation. There is a huge amount of work has happened in computer simulation. One Indian physicist from Hyderabad, Anisul Rahman, did enormous. He died, otherwise he would have got a Nobel Prize. Huge amount of work on computer simulation. And he was the first one to really simulate water. He simulate argon, many other things. And Macton Karplus who got the Nobel Prize later, did the first protein simulation on thing. So computer simulation became huge post 1970 when computers become available, particularly in 1890s. And now, of course, even your desktop has, or your laptop has tremendous power. What your laptop has now was more than what we had in 1970s in the whole Brown University. Okay. So these are they did the computer simulation of some many different things, many chemical reactions, Karplus, Levitt, and Warshall. Warshall and Levitt is the multi-stage chemical reaction, multi-state models, and they are the pioneer of QMM, quantum mechanics, molecular mechanics treatments that we use in enzyme kinetics. Martin Karplus was the first one to do protein dynamics, and I can go on telling stories about them because I know this gentleman, but I'm not going to do that. But this is simple what these guys did. I want to know how enzyme works. Enzyme is breaking a bond between the A and B. Now, if you were trying to break that bond in liquid, it will take millions of years. It's a huge barrier. But enzyme does it in seconds. So the people knew that enzyme is like magic, but people don't know what is magic, how the magic works. Combination of computer simulation and single molecule spectroscopy, quantum mechanics, statistical mechanics, all put together to explain how the magic happens and how the fluctuations of enzyme brings in. So the C moves close to the bond. Distance between the molecule and the enzyme is Q, one coordinate. Bond to be broken is another coordinate, X. This fluctuation comes close to the bond and makes the bond weak. If C has a charge, positive charge, then that comes to and withdraws the electron from the bond. And as the electron density from the bond is withdrawn, bond becomes weak. That is the magic. We talked about the magic, but we came to know of the magic only. That's why they were given Nobel Prize. Okay, and there are many theories were there. These are from Lellinger book. We didn't know what actually was happening, but now we know how it happens. That how barrier goes down in enzyme and all these things and all beautiful. There's a beautiful book by Nelson, Biological Physics. If you want to interested, a very good book, Biological Physics. And this is, I was talking that if you have milk, then is a beta galactosidase. That beta galactosidase breaks the bond, and this CO bond here on lactose that is broken to galactosidase and glucose, and that is done by this enzyme. And one studies by using a replica kind of a thing, and by single molecule spectroscopy, one finds the time scale. What is the mechanism of this beautiful experiment? That's what I'm saying you, that the amazing connection, protein folding, rugged energy landscape, your spectroscopy, your microscopy, all these things. Microscopy comes in isolating the reaction event. Spectroscopy comes in, give you the dynamics and other information. Amazing things that happen. There's a lot of agreement between these two gentlemen that hindered their Nobel Prize. Karplus and Warshall were fighting. Martin Karplus saying that dynamics is important. Warshall was saying dynamics is not important. And so I always say these things at Master versus Part 1, Star Wars, that enzyme is an active basis. So in one case on the left, is they are just one is doing without any movement. The other guy is jumping up and down. So Martin Karplus is jumping up and down, and Ariel Warshall is the left guy. So again, the electron transfer reaction, I come to it because I go to photosynthesis. There is again another controversy, Levy versus Rudy Marcus, how the electron transfer takes place. Marcus formulated theory saying that the electron transfer takes because of solvent fluctuations, and that turned out to be correct, as I told you. 2 minutes, I'll just say there are many beautiful problems that has happened because of computer simulation, because of spectroscopy and microscopy. And so the problems of whole physical chemistry, classical physical chemistry, the classical and time-honored problems of physical chemistry have undergone rebirth completely. So many things that we studied, for example, we studied ions. People have done monatomic ions for a long time, potassium, sodium, chloride. They're important, everyday ions in your body right now. And then there are nitrate, acetate, sulfate. And people earlier was working on potassium, sodium, chloride, but because of now very important discoveries in what is called two-dimensional infrared spectroscopy and computer simulation, we can do nitrate ions. So in ion, just like we Marcus say, rotation of water molecules, a potassium ion moves from one place to other, and at the same time the water facilitates it or deterts it through rotation of its dipole. But in a nitrate, the molecule, nitrate is a symmetric molecule, it rotates, and that also interacts with this. So the situation is more complex but more beautiful at the same time. And here is showing that the angle jump, the water jumping and nitrate jumping at the same time. So water jumping at RV, omega V prime, going to RV prime, omega prime, nitrate is jumping at the same time. So they are together. Sometimes they're little gap of a few femtosecond, of less than a picosecond. But these are beautiful phenomena. That's what I'm trying to say, that there you can actually understand all these beautiful, beautiful things. Then you can say, okay, nitrate is much bigger. Okay, nitrate is slightly bigger than, not much bigger, slightly bigger than, but much faster. My much was misplaced. It diffuses twice as fast as. That's the Harris-Einstein's relation, which says it's so inversely proportional. So nitrate diffusion should be less than half, but it's a more than twice, factor of 4 difference. Why? That's because of the symmetry. Nitrate ion is symmetric. So it's the energy landscape, just like a protein folding. Free energy landscape is such that it can accommodate. And then this graph, you don't have to see, just see the right one. It shows that how nitrate ion moves farther in the same time because it jumps. That's the correlation. Then there's beautiful thing of, say, ion correlation with a breakdown of potassium and chloride. How are they distributed? We never knew before. And they form these beautiful ion pairs, like we have in vortex field in physics, and breakdown, and this leads to breakdown of the screen Coulomb's law. But this is just beautiful. It's all published now. So this is the breakdown of screen Coulomb's law in this thing. So you write Coulomb's law, Q square by epsilon r. That will not work in less than 1 nanometer or 2 nanometer. But they never knew that before. We just knew theories don't work, but we didn't know why theories don't work. And there is this again, sodium nitrate ion and jumping, and many new results and dynamics. So the last part is one beautiful thing that came out from our lab is insulin. Insulin is biologically active as a monomer, but it's stored in our body in pancreas as a hexamer. Coming out of hexamer, it becomes a dimer. So there are two important things: how hexamer becomes a dimer and how becomes a monomer. I repeat, monomer is the biologically active species, but it's stored in your body as a hexamer right now. Now, when the need appears, hexamer comes out, and when it comes out of the pancreatic cell, it becomes a dimer, and dimer has to dissociate. And these are two beautiful processes that we worked on. And this is showing how dimer is formed, monomer 1 and monomer 2, yellow and green. There are 4 hydrogen bonds, backbone hydrogen bonds. And there are many interactions between hydrophobic big residues, phenylalanine, and all these things. And how does it happen? We worked it out that how the beautiful role water plays in your body right now, how it navigates a dimer. There's water. The picture on the left below panel is a beautiful picture. 5 nanometer apart, so much water molecule, but the two insulin knows each other. This is a new thing. To do or know, and they navigate. The water helps them navigate in everywhere. So that's why this paper that you wrote here actually says that is the water controls the dynamics. And many more things have been done. How the different structures, the insulin dimer, which is the state A on the right hand side of your top part one, and then how it goes through different transitions and the energy landscape, how it goes through one to the other, and how it navigates the separation. These are all new things that are when insulin hexamer. I told you that there are two beautiful things. One is this is dimer to monomer. I just told you here. Then these are all published papers, and you will be getting the copy of that. If you have a problem, then write to me or ask me questions. Hexamer is another beautiful story. This is hexamer. Why is hexamer stable? You ask the question. Then you ask the question that everybody told us, X-ray told me there's a cavity. So I immediately said, what is the cavity? And my students, Shomok and Shantan, and we collaborated with some with two biologists, Professor Gopal and Deshmukh. What is really structure of the hexamer and how many water molecules are there? You know, X-ray crystallography doesn't give you, because water, by the time you crystallize, you lose much of the water, many of the water. But we collaborated, we found that they said some number of water molecules are there. That more or less agreed. We found 2-3 more than what they found, which is expected. And how the hexamer is stabilized, because there are 2 zinc ions on the top and bottom. And then the water molecules are 3 water molecules are solvating zinc and 3 histidine. So 336 and 4 remains free, which comes as showing here with time, distance. It goes out and comes back, goes out and comes back. This is the entropy I was talking. And then dynamics of cavity water and all these things we did. But this is the beauty. This homework did. When you remove the water, the insulin collapses. It becomes a molten globule. And collapse within a few femtoseconds. You know, that is certainly is a computer simulation, and all there are other issues. In real world, will not be so fast, but still you can see the beautiful movie. Water removed and it is collapsing. Okay. And then the day it was published, we got phone call from California. Many phone calls actually. So CNN News brought it out in a very big way. Insulin falls apart without water. I should have had it. I had it in there. What they wrote actually very interesting, but that we don't have here. But you can read it. You wouldn't see insulin. If you are taking insulin, you are not supposed to drink alcohol, ethanol. So we try to find out what does it do, and it does a lot of funny stuff. It distorts the structure, as you can see, and ethanol comes in at the border and kind of screws it up. And as it screws it up, your hexamer might not become dimer. And it's a bit of a problem. Insulin and ethanol interacts very strongly. That's why you are not supposed to drink when you are taking diabetic drug. Okay. The last thing is the energy transfer in photosynthesis. As I told you, this electron transfer. Beautiful work has happened in spectroscopy, and my friend Graham Fleming and many others people. They said that this is quantum mechanics at biology. The coherent quantum state survives in biology and responsible for the fantastic efficiency of photosynthetic systems. Fantastic, you know. In a battery, we hardly get 15-20%. Nature does with 100%. A lot of beautiful work going on there. So we worked on that. Energy transfer from chlorophyll molecules to reaction center is highly efficient. Then in terms of long-lived quantum coherence. So this is the thing that we have several papers written. And so the issue is essentially the issue of Einstein, whether it is random walk or it is quantum coherence, which is localization. So again, there are several papers. We have Datta and Bagchi. And here is the example, the coherent transfer in coherent transfer. This condition between them, photosynthetic system where both coherence and quantum mechanics plays a big role. It's amazing that even in photosynthesis, the first act of life, first event of life on planet Earth, quantum plays such an important role. Okay, so these are the students which is much of my, I always keep Robert Swansea, my mentor, is a great man. Did work with him 3-4 times. And then Pooja did insulin, Pooja Shomok, Sayantan, insulin, and many other things. Pooja is the ion pair, some are older work. Rajesh did the photosynthesis. The group at some time they did last year. It has grown. Not everybody is here, but still we started. There was no StatMech in physical chemistry in India before we started. Now there is a fairly big group. And Carl Sagan, few things now, more less demanding things. The beautiful thing by quotation by Carl Sagan, you know, he's a cosmologist, One glance at a book, you hear the voice of another person. Perhaps someone did for 1,000 years. To read is to voyage through time. This is what I usually use, voyage through time. I added voyage through space also, but microscopic space. There are books written. One more big book is being written, that is timetable and relaxation. This is molecular reaction. They could just some of these. What are things we will find there? And statistical mechanics is the book. It's the first book really of this kind. Major textbook by in India, from India, statistical mechanics. And we are writing the next one, which is the time dependent stat mech. Hopefully it will be done. And that's why I wanted to tell that, you know, this book, How to Perform Well in Academia, is very, very important for a young researcher. And we often don't talk about it. There's a lot of talk of that when I was doing PhD. Here we don't talk. Nobody discusses these things too much. Or the way they talk, that does not quite reach to young researchers. So I wrote many blogs. So my students put this blog together, mostly Shomishta and Shomak. How to Perform Well in Academia, guidebook. You will like this book. It's available in Kindle, very cheap, some 50-60 rupees. And it's a good book. Many advice, many discussions, how to choose this advisor, and many other things. And there's a humorous book. This is the first one of my blog again, which is Three Donkeys of Malleshwaram. And they end with this famous poem of Rabindranath. That's what I was started in the beginning. My very first 1 minute of saying is the same thing I was saying. Path of thy creation thou hast strewn with reception. Laid down thy wave adept for the naive inept. These are communists for whom no darkness is terrible. English translation is really not that great, but Bengali is beautiful. তোমার সৃষ্টির পথ রেখেছ আকীর্ণ করি বিচিত্র ছলনা জালে হে ছলনাময়ী মিথ্যা বিশ্বাসের ফাঁদ পেতেছ নিপুণ হাতে সরল জীবনে এই প্রবঞ্চনা দিয়ে মহত্তরে করেছ চিহ্নিত তার তলে রাখুনি গোপন রাত্রি তোমার জ্যোতিষ্ক তারে যে পথ দেখায় সে তার অন্তরের পথ সে যে চির স্বচ্ছ সহজ বিশ্বাসে করে তার চির সমুজ্জ্বল path of science and research, if you are lucky and if you are industrious and hardworking and honest, is a wonderful life till now. Thank you very much for your attention. Bye. Arshad.
Thanks, Professor Bagchi, for your very nice lecture, and I think that all the listeners will be highly benefited to this lecture. And now, if there is any question from anybody, they may ask.
I didn't know I took so long. I was happily talking.
If any question, sir.
Sure, anybody can ask.
Okay, you may ask the question. One question is Mohammad Joshimuddin. What is the reason behind the folding of protein in the secondary structure, and what is the utility of this folding?
Oh, yeah, it's a very good question. There are 3 stages we identify as protein folding. One is the collapse, molecule becomes, and different proteins collapse, become smaller, tight. And during that itself, or towards the later part of the hydrophobic collapse, is the secondary structure, helices form. Helices form fast because they have tremendous propensity for the helix because of both hydrophobic contact and backbone hydrogen bonding. And then they tertiary contact, which is distant contact formation, which is slowest, which is the one actually takes much time. So secondary structure forms because of hydrogen bonding and hydrophobic interaction. And that means, see, an extended state is the hydrophobic group by phenylalanine, valine, isoleucine. They disrupt hydrogen bond network, and water doesn't like it. However, so when they come close to each other, to isoleucine comes close to each other, or to phenylalanine, they exclude the water from there, and that gives rise to a stability called hydrophobic interaction. And there's a lot of papers on that, including in my book. Hydrophobic is a big chapter in my book, hydrophobic interaction, and this has been discussed. So yes, hydrophobic interaction, hydrogen bonding, that stabilizes the secondary structure, and that also drives the formation of the secondary structure. I think that answers his question.
Okay, sir. Any more question? No. Okay, sir. Thank you. And I'm now requesting our head of the department to conclude.
Yeah,
this session.
you should have told me. You should have told me that time is running out. I have happily went on talking.
No, it's not. And sir, one thing is that mainly the hydrogen bonding. It is the main responsible factor for all these types of biomolecule interaction. Is it?
Yes, to a great extent. It's amazing how much hydrogen bonding plays a role. It is because there are many of them. Like the insulin dimer, insulin dimer is largely stabilized by 4 hydrogen bonds, backbone, hydrogen bonds. There are hydrophobic interaction, but hydrophobic interaction breaks fast. Hydrogen bond breaks last. Hydrogen bonds itself is probably about twice or about that more interaction than if I can define hydrophobic interaction. Hydrogen bond is a little bit stronger, but more than that, there are many hydrogen bonds. Whether you look at DNA, whether you look at protein, there is a lipid, there is abundance of hydrogen bond, and of course water is there, and water is the real hydrogen bond forming liquid. So this is really amazing how this is, it's famous by John Mary Lane, another Nobel laureate, who built this molecular architecture, and he has huge volumes.
বাবা আচ্ছা ঠিক আছে ওকে So there are another question in the chat box.
Sure, tell me.
Why is Aristotle so celebrated despite of the fact that his contribution to science is not that great?
Aristotle is celebrated because you have to understand he was born 384 BC, and he established this modern school of philosophy. He wrote extensively. He kind of one of the very early guys who pointed us to the questions of. He traveled far and wide. He came all the way to Egypt and far. He made one of the first maps of the world. He asked questions. Yes, he got the answer many times wrong, but he was a great intellectual who established a great school, who wrote widely, and he interacted with people. He almost was a revolution himself. Remember that Aristotle was a student of Plato. The Plato school, you have seen the great painting by Raphael, right? The School of Athens. Please see that painting. School of Athens, famous painting. And just wonderful. And Plato was a favorite student of Socrates. So this trio and many others, of course, are the great Greek school. So I would give Aristotle credit for, and he has just many other things on philosophy, then books on the formation of nations, how the government should be formed. So Plato and Aristotle are, that's why given credit for the first guys who in writing available. I'm sure in India also people thought of these things, and probably, but we, India, unfortunately, we did not write much, or whatever we didn't survive. Egypt also did not write too much. That amazing. Though Alexandria Library in Alexandria has some old documents, but nothing like preserved well like that of Plato and Aristotle. And Plato wrote about Socrates. So you know about Socrates because of Plato.
Sir.
Sure.
Any question?
Sir, a lot of like scientists were demoted from the credit they deserved because of the fact that Aristotle suggested some kind of theory which was not at all correct. this happened with the structure of solar system too.
Right. Correct. See, we are here not to give credit to people after 2,500 years. The fact that you have to be very kind and generous to people. Biggest mistake we make being very critical of people. That I would imagine is a human vice. But whole world holds Aristotle and Plato and Socrates in great respect. Yes, they are wrong many places. Aristotle is wrong, but Aristotle was the first one to write down about these things. He probably was among the first ones who asked the questions. See, there is credit goes on asking the question. You have to understand, most of the people, if they walk, you can see them walking. Very few. All look at the roads. They don't look at the sky. So there are few of us who look at the sky and ask questions. And I think we have to respect them for that. They do fall in the ditch and die, and they make mistakes. That's what Rabindranath's point is. Tomar Srishtir Path Rekhecho Akirino Kori. These are one of the dangers Rabindranath talked about.
Okay, sir. Thank you, sir.
Sure.
I'm now requesting Arshad to speak something.
Thank you, sir, Bagchi sir, for your wonderful and nice lecture. And specifically, personally, I enjoyed a lot, and it was very much informative for beginner and basically for students. And I
Thank
see
you.
from the comments, most of the participants, they appreciated much, and they mentioned that they have enjoyed. It was very informative. So thank you very much, sir. And I will be happy in near future again if, sir, you give us some time. Maybe we will arrange some other programs also in near future.
Sure.
And,
Thank you. I can give a nice talk on physics and chemistry of drug discovery. That is our work on insulin, but other things are also made for me, and many other things that we have done. That could be a good topic when you can fit in. Give me a break of a month or so, and after 2 or then you ask me, and I'll be very happy, because when students are there.
Yes, sir.
You know, two things. I love doing that, interacting with students. I wrote those books for students. Second,
yes.
As a national chair professor, my mandate also.
Yes, sir.
You know, a national professor must reach out to everybody.
Yes, sir. So it would be nice. We will contact you after some times once again.
Absolutely. I enjoy it.
Okay,
Talking
sir.
with you guys. Thank you very much.
Thank you very much, sir. So now I request our HOD, sir, and chairman of this session to give the concluding remarks. Sir, please.
Thank you very much, sir, for your delightful and electrifying lecture.
Thank you. I'm sorry it became long. I did not even sitting in my room happily, and I did not look at the watch at all.
Oh, okay, sir. So actually, we are also grateful to all the audience for their presence, and I think they will be highly benefited from the lecture. So, sir, in near future, if you arrange some more lectures, then we expect that you will extend your kind cooperation. Actually, sir.
Yes, sure.
So actually, you have something told to Arshad. So accordingly, we will arrange something after 2 to 3 months. Okay. Sir.
Thank you.
Thank you again, sir.
Okay then. All right, we are done, Arshad.
Oh, yes, sir. Okay, thank you very much, sir. So let's conclude for today.
Okay, thank you.
And another issue, sir, I also will be happy if you discuss on how to, develop in academics for our students or scholars. Maybe in some
you can arrange that talk. That my 2 books.
Yes, sir. Yeah.
Particularly this book.
Yes.
This book I told you available in Kindle is we just gave it very cheap, some 50 rupees or so.
Yes.
But I can discuss a little bit because this is my favorite topic, and Indian National Science Academy is supposed to bring out this book in a better way. But this is something I think is must be discussed. I
Yes,
think
sir.
really some frank, transparent way. Our students, we should be told.
Yes.
As I told in this book, sometimes students get exploited. So students need to know how to handle situations. That's what I talked in this book. How to select an advisor, how to select a problem, what
Yeah,
is
that's
required
nice.
to be successful.
So I see Dr. Minal Pal, principal scientist of CGCRI here, participated in this program. So Minalda, if you want to say something.
No, actually, I have heard several times Professor Bagchi on different occasions, and I really like his all the deliberation. It is really nice, and he explained all the things to the students as well as to new faculty in such a way. I think everybody can understand very easily. So, from my side, Professor Bagchi, many thanks, and I will also expect in near future, if possible, I will arrange some some lectures in our place. It is Central Glass Ceramic Research Institute, Kolkata. You know very well.
Sure.
So please respond to my call in future.
Sure. Thank you.
Thank you,
Thank
Professor.
you. Thank you. I will. Thank you.
Okay, sir. So thank you very much once again. Thank you
Okay.
all. So let's conclude the program.
All
Okay.
right. Bye then.
Thank you.
Bye.
Yeah.
Stay safe.
Thank you,
Yes,
everybody.
bye, sir.
Okay, sir.
Thank you, everybody.
Okay,
Bye.
okay,
Bye.
thank you.
Okay, thank you, sir. Thank
Yeah,
you.
thank you, everybody.
Okay, sir.